I'm an evolutionary biologist, and I'm most interested in what kind of things can evolve and what kind of things cannot. If we had an answer to that question, we could know something about our evolutionary futures. And if you look at certain features like bilateral symmetry, for example, if you take a line from our head through our tail, we have a left and a right-hand side, and they're symmetrical, they're mirror images of one another. That hasn't changed for 540 million years, an incredibly long time. On the other hand, size, shape, color, do you swim, do you run, do you fly, those things can evolve pretty quickly. And so why can some things evolve and others cannot? So we found a protein, beta-2 tubulin, which makes sperm tails, which hasn't evolved in 60 million years. And every time we change it just a tiny bit, the sperm falls apart. So how did this protein evolve, in a sense, into a corner which the fly seems to be stuck with? That's our question. Things we're stuck with, can't participate in adaptation. How do we get around that? How can these slow evolving things kind of get unstuck? The way that I study this is to find an alternative version of this protein, in this case the human version of the protein, and ask, can it replace the fruit fly version of the protein? We do this by expressing the human gene in the fruit fly and ask, does it work? If it works, we know, oh, another version of this protein can function. So there are choices. Nature just keeps picking the same one, the one that the fly currently has. It's like the New England Patriots of proteins. It keeps winning competitions. But there's an alternative. If the human protein doesn't work, that tells us maybe, as our previous studies have found, beta-2 is the only way for a fly to make a sperm tail. And then the question is, well, how did it get there in the first place? Well, something else must change in the sperm tail that allows beta-2 to change, and that's the coevolutionary hypothesis. There's a medical relevance in addition to an evolutionary relevance to this research because humans use the same beta-tubulin in brain development as they use in sperm development. So if the human version of this protein works in the fruit fly, we can use the fruit fly as a model to study human tubulin mutations that are impactful on brain development. I find the world a fascinating place. I think it's incredible when you just take a walk through the woods and see the diversity of life that you find around you. And I can't help but ask, why? Why does it look the way it does? Why do so many things look similar to one another, yet there's so much variation at the same time? This is research to answer that question at a most nut and bolt atomic level. But the answers apply so broadly if we could find a generalization that coevolution is the key to unsticking a difficult phenotype. Or no, you don't have to wait for something else to change. There are just fewer choices available, and so it takes longer for nature to try them all, so to speak, before it finds another choice that might provide a different quality of sperm. If we could understand that, again, we may have some sense of where our evolutionary futures go.